Hi everyone, today we are going to talk about riboswitches in bacteria, an important element in RNA structure. In this presentation we will look at the following aspects. What is a riboswitch and how did it evolve? The classification of a riboswitch, the structure and function of a riboswitch, the role riboswitches play in transcription and translation, and finally we will discuss the importance and applications of riboswitches. So what is a riboswitch? A riboswitch is a region of RNA in bacteria that combines small molecules at the 5' untranslated region. This binding process determines whether or not the associated genes are expressed. In other words, this RNA element is involved in the regulation of gene expression in bacterial cells. Here you can see an up-to-date table showing the families of riboswitches determined by the ligand they are binded to, followed by their classification, determined by the secondary structure of their aptamer domain. Riboswitches were first discovered through an experiment on vitamin B derivatives, and since have been found to occur naturally as molecule binders. They were discovered only in 2002 in bacteria. Today there is approximately 10 to 12 well-known classes of riboswitches discovered to date.
Hi, I'm Michelle, and I'm going to talk about riboswitch structure and function. The riboswitch has two main sections, the aptamer and expression platform. Riboswitches can be broken into two classes based on their structure, those with pseudoknot folds and those with helical junctions. The pseudoknot is formed by base pairing between a loop of RNA and outside region. Junctional riboswitches combine several helices by stacking them on top of each other and packing them side to side to make the commonly seen Y shape. When a metabolite is in excess in the cell, it binds to the riboswitch, which will stop its production. A riboswitch is not always a single unit. There can be two sensor domains or two whole riboswitches right next to each other, which is present in the glycine riboswitch. Although it may seem like the aptamer and expression platform are two separate structures because of their different functions, they are connected. They overlap each other in a space that is called the switching sequence. Here, the information can be passed from the aptamer to expression platform domain. The conformation of the expression platform will be decided. The aptamer is a specialized ligand receptor. It can range in size from 70 to 200 nucleotides. The types of compounds the aptamers bind are small purines, coenzymes, amino acids, and metal cations. Each aptamer is very specific to the ligand it can bind. Small variations of the compound may interrupt binding. The aptamer has several different ways of ensuring it only selects the correct metabolite. Most aptamers have a tight ligand binding pocket and hold in the ligand with van der Waals forces. Hydrogen bonding between ligand edges and non-paired nucleotides of RNA is used to ensure the correct orientation of the metabolite. As seen here, only the one correct metabolite can bind the aptamer. The aptamer then signals the switching sequence and expression platform to change conformation to an on or off position. The expression platform is a secondary switch which dictates whether gene expression will be turned on or off. A ligand is bound to the aptamer which signals folding in the switching sequence, and that will cause the expression platform to fold into the designated conformation. The conformation will determine whether transcription, translation, or other gene expressions are terminated or continue to carry on. The conformation of the expression platform will make the ribosome binding site available or not for translation, and the platform can fold into either a terminator or anti-terminator position for transcription. This is a simplified version, as the 3D folds of a riboswitch are very difficult to determine and visualize.
Hi, I'm Natalia, and I'm going to talk about transcription and how riboswitches affect this process. What is transcription? Transcription is the process of converting DNA to RNA. This process is a key step in order to transfer genetic information to the ribosome. Each gene can be transcribed at different rates and efficiencies. RNA differs largely from DNA in that it is single-stranded and often contains short sequences that are complementary to other sequences that allow it to fold over on itself. Riboswitches regulate transcription. As we know, riboswitches regulate gene expression almost exclusively in prokaryotes. Riboswitches control gene expression by promoting innate transcription termination or by impeding translation initiation. We also now know that riboswitches can regulate the ability of RNA-dependent helicase row to terminate transcription. Riboswitches promote transcription when there is no ligand present and the anti-terminator loop is engaged. They terminate transcription when a ligand is present and attaches to the riboswitch. This causes a terminator loop to form, which does not allow RNA polymerase to attach to the expression platform.
This video was made to demonstrate how riboswitches have the ability to terminate transcription. So present here, we have an mRNA strand with our 5' and 3' ends. And as described in the previous section, we have an aptamer region and an expression platform making up a riboswitch. In the aptamer region, we have two loops. One is described as the anti-terminator loop. This is engaged when there is no ligand present, and RNA polymerase can easily bind to the expression platform and continue transcription. However, when a ligand is present, it binds to the RNA and changes the riboswitch shape, allowing a terminator loop to form, which stops RNA polymerase from engaging with the expression platform and hence terminating transcription.
Hi, I'm Haya, and in this video I will describe the translation of a riboswitch.
So, as we know, translation is the second mechanism after the transcribing DNA to a single strand of RNA. This RNA then requires a ribosome to be translated into a protein. For translation initiation, which occurs in prokaryotes when a specific ligand binds to the aptamer domain, as shown in the picture, this causes a rearrangement in the structure of expression platform site, which includes the sequencing of the Shine-Dalgarno binding site, SD, proposed by the two Australian scientists John Shine and Dalgarno, or known as a ribosome binding site, RBS, where a ribosome should attach to this site and start the translation of RNA, as shown in this picture. The SD site is located 8 base pair upstream of the sought site. In initiation, the RBS is already not free as shown. It's bonded to the mRNA. As the ligand binds to the conformation is changed, and the RBS breaks bond from the RNA and can be accessed by the ribosome and start the translation process to the stop codon. In this slide, a lysine riboswitch will be an example of an initiation translation where the gene is inhibited by the lysine ligand and the gene will not be expressed to a protein due to the end of the hairpin loop that is formed. It's known to be one of the most common riboswitches, an element that controls the expression of a lysine C gene. In translation termination, it stops the mRNA from producing the protein due to the efficient amount it is produced in the bacteria. As you can see, in order to stop or terminate the translation mechanism, a specific ligand must bind to the aptamer domain, and this changes the conformation of the RNA, which then hides the ribosome binding site RBS or the SD as shown in the first slide, through the bonding between the RNA and site. As a result, translation is then terminated and the mRNA will not stop producing the protein.
Hi, my name is Samira ElMaadi and I will be discussing the importance and application of riboswitches. Riboswitches play a role in gene expression in RNA. Research has shown that they are responsible for a decade worth of gene control mysteries. Recent studies have found numerous new RNA motifs and non-coding RNA in bacteria. This means more riboswitches could be identified. Riboswitches control the expression of 4% of genes in bacteria. They are also found in eukaryotes and archaea. They are competing with protein in gene expression and are successful in being accurate and highly sensitive. Riboswitches have the potential to be used as drug targets. We now know that riboswitches are the drug target in a number of drugs. That mechanism was unknown. Riboswitches can distinguish between different cognate molecules as well as their existence in bacteria. The use of riboswitches as antimicrobial drug targets has many advantages over classic antibiotics. They have a lower toxicity because riboswitches are not found in humans. They are controlled by small compounds which are simple metabolites. This makes them easy to deliver into the system. This would make them cheaper to manufacture and would make them easier to modify. There is also less chance of bacterial resistance due to the fact there is only a single type of riboswitch in bacterial genes. This makes the mutation harder to stop the effect of the antibiotic. Thanks for watching.